Psychology in the Fast Lane, Difficult Topics Explained. Elements of Sound Methodology. Have you ever had your palm read or your fortune told by a psychic? These are examples of pseudoscience. Pseudoscience consists of claims based on little to no scientific evidence. These claims are usually blindly accepted as fact or passed down as an old wives' tale, but in reality, the claims have never been tested. The scientific method provides a framework for true science and sound methodology to occur, and is a set of procedures and attitudes that guides us in asking questions, gathering evidence, and drawing conclusions. At the heart of the scientific method is empirical evidence, evidence that is observable and measurable. Unlike pseudoscientific claims, you can verify that empirical evidence is true and accurate. Maybe you've heard the saying, "An apple a day keeps the doctor away." Is this claim pseudoscience, or is there empirical evidence you could collect to test this claim? According to the scientific method, you must formulate a hypothesis, which is a specifically stated and testable prediction. In this example, your hypothesis is that eating an apple every day will keep the doctor away. But before you can design a study, you'll need to operationally define your hypothesis. In other words, specify exactly how the variables in your study will be observed and measured. For example, your operational definition could be that a Macintosh apple eaten each day will lower blood pressure or reduce the number of times a person goes to the doctor in a year. When you design a study, you must choose from one or more of 5 research methods: observation, survey, case study, correlation, and experimentation. Which method you select depends on how you operationally define your hypothesis and other factors such as the number of subjects that are available, or if you're measuring cause and effect. After you collect your empirical evidence, you'll analyze your data, draw conclusions, and report your findings. Psychology in the Fast Lane, Difficult Topics Explained. Experimental Design. Think of the experimental method as a way of isolating what is causing something to happen. Here's a simple example of isolating a cause. You notice that your car is making a squeaking noise when it's running. You walk around the car and listen for where your sound is coming from. You notice that one of the parts is wiggling around. By listening to locate the source of the sound, you used your auditory perception, and you've confirmed it with visual perception. So you hold it in place and the squeaking stops. You let it go and the squeaking continues. After repeating this action, you can conclude that the wiggling part is the cause of the sound. Tighten up that part and you now have a quiet car. First you observed the squeaking sound, then you described it by pinpointing its location. Then you made a prediction, or you hypothesized that the wiggling part may be related to the squeaking sound. Then you tested your hypothesis by stopping the wiggling part and observing the sound stop. You repeated the test and compared the non-wiggling and wiggling conditions, or the control and experimental conditions. Then you came to a conclusion: the wiggling of the part is causing the squeaking noise. And you acted, tightening the mechanism to keep it from wiggling, and you fixed the problem. Science at its most basic. Now, let's look at an example from the psychology field. A group of neuroscientists observed that people with traumatic brain injury, which is severe brain damage caused by physical accidents, have difficulty recognizing facial emotions very well. They formed a hypothesis that TBI leads to impaired recognition of emotion in faces. Their solution was to create a group exactly equal to the TBI group in every way except having TBI. Same gender, sociocultural, and age characteristics, just not brain damaged. Then they tested each group by showing them pictures of faces and asked them to identify the emotions in the pictures. They found that the TBI group did in fact recognize fewer emotions accurately, an average of 15 out of 20, compared with the control group's 18. They concluded that TBI does cause impairments in recognizing facial emotion. Just remember, the experimental method is designed to determine the cause of something by isolating the hypothesized causes and comparing their controlled results. A quick note on cause and effect. Cause and effect are at the heart of experimental science. The examples in the experimental design video illustrated cause and effects. Notice that the procedures were the same. Two conditions have to be in place so that one can be compared with the other. The first example compared the car's sound with a hand on the wiggling part to the car's sound with no hand on the part. In the second example, we compared facial emotion recognition between those with TBI and those without TBI. The conditions can be arbitrarily chosen and assigned. They are said to be independent of anything except chance or the researcher's choice. However, in the TBI example, the TBI patients didn't have a choice to be in the TBI group. They were in it because they had an irreversible condition that could not be randomly assigned. They had to be compared with a matched control group. In other studies, a group of people can be split into two or more groups, with one group serving as a comparison or control group. Think of a study where one group gets a drug and the other gets an inert substance, a placebo, with the groups formed by the flip of a coin. That is random assignment to levels of an independent variable, which is the presence or absence of the drug. Presence or absence of the drug is expected to have an effect on another measurable variable, an outcome variable, for instance, the amount of pain experienced after taking the drug. Since differences in that outcome variable depend on differences between the groups, the outcome variable is called the dependent variable. In a nutshell, independent is to dependent as cause is to effect. Another way of thinking about it is that cause happens before effects. You have to do something first to get a result. Bypass Publishing presents Psychology in the Fast Lane, Difficult Topics Explained. Independent versus dependent variables. Correlational studies are unstructured observations. You are simply recording what you can observe. Experimental methods require structured observation that results in the manipulation of variables. The term variables simply refers to something that varies. In this case, we need to discriminate between two types of variables, independent and dependent variables. A dependent variable is something that we want to measure as an outcome. In an experiment, we manipulate the independent variable by creating a control condition, which is basically no manipulation, and an experimental condition, where we have manipulated something. In both conditions, we measure the dependent variable to determine if there's a difference in observed outcomes. So as you can see, the outcome variable y is dependent on variable x, because as we manipulated x, the outcome y was affected. Okay, by now you should be familiar with the procedure and vocabulary involved in an experiment. Terms such as trial, random assignment, control group, experimental group, and independent and dependent variables should be second nature to you. And you should be aware that an experiment is designed to determine the effect of an independent variable on a dependent variable. See Difficult Topics video, Experimental Design. Now, in a perfect experiment, only the independent variable would have any effect on the dependent variable. But alas, life and experiments are rarely perfect. Extraneous variables are often present in an experiment and may or may not compromise the validity of the experiment. An extraneous variable that affects the validity of an experiment is referred to as a confounding variable. Now, to better understand what I mean, let's take an experiment that's been done in grade schools for generations. Let's see whether sunlight makes plants grow. You start your light growth experiment by placing one plant in the sunlight and one in the darkness, and then you wait. When you see that one of your plants is doing better than the other, you automatically assume that it's due to the sunlight. But you may be wrong. Did you accurately measure the water given to each? Or how about the soil and air temperature, or the size of the containers, and even the location? These are all extraneous variables, extra variables that may or may not interfere with your experiment. Now, if any of these extraneous variables impact your results, then we call it a confounding variable. Think of all the extraneous and confounding variables that may be present in a psychological experiment, especially those involving humans. Then you'll get an idea of why it's so important to recognize extraneous variables, especially the confounding ones. Bypass Publishing presents Difficult Topics Explained: Psychology in the Fast Lane. The brain functions. Humans are different from other animals. We exhibit advanced behaviors such as verbal communication, and this has allowed us to create sophisticated art and civilization. If we look at other animals, bears are stronger than we are, dogs can hear sounds and smell odors that we can't, and an eagle can see farther than we can. Well, what makes us special then? To answer this question, we have to look at the human brain and compare it to the brains of other species. Now, judging from this picture showing the sizes of various animal brains, it first appears that bigger is better. However, this is a generalization that requires further inspection. If it is only size that matters, then humans would display less sophistication in terms of behavior and abilities than an elephant or whale, yet we know this is not the case. For this reason, a more meaningful approach is to examine the brain volume with respect to different brain structures. As we know, the brain is divided into 3 general levels of organization: hindbrain, midbrain, and forebrain. In general, the hindbrain directly connects the spinal cord to the brain and is responsible for life support functions such as breathing, heart rate, and blood pressure. The midbrain is associated with relaying sensory information to the forebrain, which controls cognition and motor function. Look at what changes as we look across different animal species. All animals need life support, so the hindbrain appears to be relatively similar proportionately across species. Likewise, the size of the midbrain, colored green, varies a bit by species, but generally remains the same. What appears to change most drastically across species is the size of the cerebrum, which is the part of the forebrain. As previously discussed, the forebrain is the most evolutionarily advanced because it controls higher-order cognitive processing, such as planning and problem-solving, that makes humans the most advanced animals on Earth. One factor that is noticeable from an examination of the mammalian brain is that surface area increases quite dramatically from less advanced animals, such as rats, to humans. Surface area increases via folding of tissues into sulci that allows larger cortical mass without a corresponding increase in head size. Increased neocortical or cerebral mass is correlated with a greater number of neurons, different neurons, and increased information processing power. Thus, it is the increase in the surface area of the cerebrum that makes humans more evolutionarily advanced than other animals. Bypass Publishing presents Psychology in the Fast Lane: Difficult Topics Explained. Brain Structures. Now, we all have a brain, and we all use them in different ways, but they're all divided the same way, into the hindbrain, the midbrain, and the forebrain. As you can see, the spinal cord directly connects with the hindbrain, which contains a number of important structures. The medulla is the physical connection between the brain and the spinal cord, and it's responsible for life functions, such as breathing, heart rate, and blood pressure. If this structure is damaged, you might have difficulty breathing and regulating your heart rate. In fact, damage to it usually results in death. The rounded section above and to the front of the medulla is the pons, which is Latin for the bridge. And this structure serves as a bridge between the medulla and the higher brain structures. Slightly behind it, we can see the reticular formation, which plays a role in alerting higher brain structures about incoming information. The reticular formation is involved in arousal, sleep, and attention. If we didn't know when it was necessary to sleep and what information to pay attention to, we couldn't function very well. The last hindbrain structure I'll discuss is the cerebellum. It looks a little bit like a cauliflower, and it's located behind the brain stem that connects the brain to the spinal cord. The cerebellum is essential for coordinating motor movements such as walking or riding a bike. Damage to the cerebellum could impair motor coordination so that something as simple as walking could become difficult. And now we've got the midbrain. Because it's less specialized and plays a less important role in the human brain relative to other animals, I'll only bring up two structures here, the superior colliculus and the inferior colliculus. Based on those names, it sounds like one is more important than the other, but that's not the case. Superior and inferior refer to the anatomical locations, where superior is physically above the inferior. The superior colliculus allows us to locate an object in space, while the inferior colliculus controls our ability to localize sound. So now that just leaves us with the forebrain. Because the forebrain is essential for our most advanced, high-level behaviors, we need to take a closer look here. As you can see, the forebrain is made up of the thalamus, hypothalamus, limbic system, which includes the amygdala, and the hippocampus, and the cerebrum. The thalamus serves as a relay station for all our sensory information. For visual information to be processed, a specific area of the thalamus must direct it to the appropriate place in the cerebral cortex. The same goes for auditory information, as well as gustatory, which is taste, and proprioception, which is touch. But olfaction, which is smell, is different because it doesn't get routed through the thalamus. Directly below the thalamus is the hypothalamus, which regulates motivated behaviors such as eating, drinking, sex, and temperature regulation. Regulation of these things is important because if you eat too little, you'll starve, and if you eat too much, you'll become obese. And both of these conditions are bad, so the hypothalamus helps us maintain the optimal levels. Now, let's talk about the limbic system. This group of structures includes the hippocampus and the amygdala, and it is generally responsible for regulating emotion and memory. The hippocampus plays a role in our ability to create new memories, while the amygdala is important in processing emotional information. Lastly, I want to discuss the organization of the cerebral cortex. The occipital lobe is located at the back of the brain, and it processes visual information. In front of it, we have the parietal lobe that processes sensory information from the body, such as touch, temperature, and pressure. The central sulcus separates the parietal lobe from the frontal lobe. And likewise, the lateral sulcus separates the temporal lobe from the parietal and frontal lobes. Bypass Publishing presents Psychology in the Fast Lane, Difficult Topics Explained. Neural Communication, Synaptic Transmission. Considering that any given cell might have 1,500 synapses with other cells, the reality of neural communication is rather complex, but we'll start with a single synapse, where two cells communicate. The process of synaptic transmission is simply the conveyance of information across the space between cells. The transmission part is the conveyance of information. The synapse is the space between cells. So the individual elements that we need to understand here are the two communicating cells and the space between them. The two cells are called the presynaptic cell and the postsynaptic cell. The space, of course, is called the synapse. And it's not really empty space, but it's filled with fluid and chemicals. Now, the presynaptic cell is the one that's transmitting the message, and it houses structures and processes that allow it to send a signal. The postsynaptic cell is the one that's receiving the message, and it houses structures and functions that allow it to respond to a signal. Now at the synapse, the axon terminal of the presynaptic cell lines up to the dendrite of the postsynaptic cell. So in order to communicate, the cell must make a chemical transmitter, or a neurotransmitter. There's a separate podcast specifically about neurotransmitters. Now, usually the making of a neurotransmitter is accomplished in either the cell body or in the axon terminal itself. This decision is constituted in the action potential. At the moment of action potential generation, the synaptic cell has essentially made the decision to communicate with the postsynaptic cell. The membrane of the synaptic cell contains a specific type of switch that is sensitive to the action potential and cranks up the release machinery. They only open when the action potential threshold is reached. Now, within the synapse, not a lot really happens. However, it's important to keep in mind that the action is all basic diffusion. As your high school chemistry teacher would explain, diffusion is just the movement of ions down a concentration gradient. So when the presynaptic cell releases the chemical, it's highly concentrated, and the individual molecules don't like to be so close, so as they go along, they spread out from one another. The first thing that's got to happen when the chemical makes it over is that it has to find a receptor or a target that is sensitive to it. Receptors receive this chemical message, and they essentially act as locks, and they're activated by their keys, which is the chemicals in the message. When the neurotransmitter is bound, the chemical shape of the receptor is altered, and then the receptor initiates another process that changes the state of the postsynaptic cell, which is how the message is received by the postsynaptic cell. Now keep in mind, one single neurotransmitter molecule can potentially cause wildly different changes to occur in each different postsynaptic cell. The effect that the neurotransmitter molecule has on the postsynaptic cell is all based on the reaction of the receptor. After the message is received, the neurotransmitter molecules basically pop off the receptor's binding sites and begin floating around in the synapse again. Sometimes they find another binding site, or sometimes the neurotransmitter molecules are pumped back into the presynaptic cell through a process called reuptake. Sometimes neurotransmitters are degraded by enzymes floating around in the synapse. But as far as the effects that the chemical message has on the postsynaptic cell, like I said, it depends on the receptor. Now from there, the nitty-gritty details get really complicated and honestly go far beyond what our discussion requires today. But for now, we can simplify all of the effects that one cell can have on another by categorizing their ultimate influence as either excitatory or inhibitory. Excitatory effects are those that increase the probability that the postsynaptic cell will fire an action potential. Inhibitory effects are those that decrease the probability. And note that when the current through the receptor is positive, the resulting change is an excitatory result. When the current is negative, the resulting change is inhibitory. Bypass Publishing presents Psychology in the Fast Lane. Difficult topics explained. Action potentials and axonal transmission. Now, before going through this video, you might want to view the previous video about the general process of synaptic transmission, because we're going to get a little bit more specific in this one. A potential is simply the voltage difference across 2 points in space. The action potential of a cell is the one that occurs when the cell is actively transmitting information down the axon, from the cell body to the axon terminal. The action potential is thus the voltage difference between the inside and the outside of the cell, as it transmits information along the axon. The decision to send that information is made whenever the membrane potential of a resting cell breaches the action potential threshold. Neurons normally rest at about -70 millivolts, and an action potential is generated whenever excitatory input drives the voltage difference up to about -55 millivolts. So let's run through the process chronologically, from the start of an action potential all the way through the transmission down the axon to its arrival at the axon terminal. First, excitatory postsynaptic potentials will slowly ramp up the cell's membrane potential. The cell will rest at -70, but every excitatory signal at the dendrites drives that value upward. At -70, the sodium channels are closed. However, their little switches get thrown in a serious fashion at -55. It turns out that the first part of the cell membrane that contains a lot of these sodium channels is the axon hillock, where the axon leaves the cell body. If the potential reaches negative 55 millivolts at the hillock, then the action potential will form. The sodium channels in the hillock open up and they let sodium through. Sodium is highly concentrated outside the cell, and there is very little sodium on the inside. As more sodium comes into the cell, more sodium channel switches are thrown. As more sodium channel switches are thrown, more sodium comes into the cell. This positive feedback loop creates an explosive situation. The action potential screams down the length of the axon in a flash. In fact, the entire action potential process lasts less than 1 millisecond. Now for the potassium channels. These channels are also switched by voltage, but they require a much higher membrane potential to become active. Only after sodium entry has pushed the potential way up into positive territory do these potassium channels switch open. Potassium is highly concentrated on the inside of the cell, so as soon as potassium channels are open, potassium rushes out of the cell. When all of these positive potassiums rush out of the cell membrane potential then comes back down towards the resting potential. However, so much potassium gets through the open channels that the potential actually runs past the resting potential down to negative 75 or negative 80 millivolts. This brief period is called an after hyperpolarization because it happens just after the action potential. Another interesting point about action potentials is that they're characterized by the all or none principle. Any crossing of the threshold will suffice. Like if a cell is stimulated and the action potential threshold is not crossed, then absolutely no action potential will be generated. The initial sodium channels open up only if the threshold is crossed, and once it's crossed, you get all of an action potential. If it's not, you get none. There's no such thing as a partial action potential. Bypass Publishing presents Psychology in the Fast Lane. Difficult topics explained. Divisions, systems, and structures of the nervous system. The most basic structure of the nervous system is the neuron. Here's a chart of the divisions found within the nervous system, all functioning together simultaneously. We can split the nervous system into two major systems, the peripheral and the central. The peripheral system can be divided into two subsystems, the autonomic and somatic systems. And the autonomic system divides even further into the sympathetic and parasympathetic divisions. And within the somatic system are afferent neurons and efferent neurons. Now the two divisions of the central nervous system are the spinal cord and the brain. The spinal cord can be divided into the types of neurons and their functions, but we can save that for another day. Right now, let's turn to the brain. Various researchers have suggested different ways of dividing the brain, so if what I describe isn't exactly what you remember, do some research to see why. But for now, let's divide the brain into three sections based on their location: the hindbrain, midbrain, and forebrain. The hindbrain is responsible for basic life functions and can be divided into the pons, medulla, and cerebellum. And we could make more divisions within the midbrain, but for now let's wait. Now the forebrain can divide into the thalamus, the hypothalamus, the limbic system, the corpus callosum, and the cerebral cortex. Now, part of the point of this is that there are always more divisions that can be made, but this basic outline will give you a great start to understanding the general structures of the nervous system. You can study as deep as you want by looking up the further divisions. How much of my IQ can be attributed to my genes? Now, for any one individual, this question is unanswerable. We can really only talk about heritability of IQ, which answers a slightly different question. How well can people's IQ differences be predicted by their genetic differences? For our purposes, behavioral genetics comes down to the question of how our DNA influences our psychology. You'll hear debates over nature versus nurture or genes versus environment. However, any attempt to arrive at a clean dichotomy between the forces of nature and nurture will obscure the truth about the roles of our genes and our environments. The truth is that they're dynamically interactive. Our genes influence how the environment treats us, and our environments influence how our genes work. And the relative influence of genetics and environment can never be known for any single behavior of any single individual. Behavioral geneticists use measures of variance to estimate the impact of genetic inheritance. The heritability coefficient is a number that is a mathematical expression of a proportion or fraction of variance. For the heritability coefficient, the numerator of the fraction is variance in behavior. The denominator is variance in genetic makeup. So the heritability coefficient answers the very specific question, how much of the difference that people show in this behavior can be explained by differences in genetics? Notice that this question does not address the behavior of any individual person. We're talking about variance, so we're talking about differences between people. Each tiny piece of data contains two numbers and is derived from two people. Two people differ in the behavior and they differ in the genes. We record that pair and we observe another pair of people and then another and so on. And at the end of the day, we have an estimate of how the magnitude of the difference in behavior is related to the magnitude of the difference in the genes. Well, let's take a look at a couple of idealized graphs to illustrate the point. We've laid out the relationships between two variables. Along the x-axis, we have magnitude of difference in genetics, and along the y-axis, we have magnitude of difference in behavior. We usually measure the difference in genetics through difference in alleles at selected points in the genome. We can put any behavior we choose on the y-axis, but just for fun, let's use artistic talent. In order to get the points on the graph, we need to measure differences in each variable. So each point on the graph actually represents two people and displays their difference in genetics and difference in artistic talent. In the idealized graph, I've made two circles. The one at the lower left might represent my brother and me. We don't differ very much on genetics because we're low on the x-axis, and we don't differ much in our artistic talent, which is why we're low on the y-axis. The upper circle might represent a perfect stranger and me. We would be maximally different in our genetics, being high on the x-axis, and maximally different in our artistic talent, being high on the y-axis. A graph like this indicates a large degree of heritability. Again, just to remind you, it's an idealized graph and it's not real data. Now, note that the x-axis says nothing about the actual magnitude of genes, only the difference, and the same for the y-axis. So my brother and I might both be very bad artists, but we might both be very good artists. The graph simply indicates that we don't differ much wherever we are. So here's a graph that's fundamentally impossible. This graph represents the magnitude of genes and a magnitude of artistic talent for individual cases. So each dot is a single person instead of a pair. This is the data you would need to have in order to answer our original question about the power of genes for any individual. However, these data are impossible to acquire. We all have the same number of genes or magnitude of genetic material, so there's no way to gather information on a range of the x-axis. It's possible to measure the differences between people, but it's impossible to measure the amount of genes in any single individual. So we cannot use heritability to answer a question about the individual. Knowing how genetic differences predict behavioral differences is a wonderful endeavor, even for things as controversial as IQ. We run into serious trouble when the legitimate data on prediction are twisted into unsupported claims about causation. Bypass Publishing presents Psychology in the Fast Lane. Difficult topics explained. Information processing in the brain. Sensation and perception are complementary processes that allow us to experience the world. The terms sensation and perception are often used interchangeably, but you'll soon see that they're distinct processes. Sensation is a passive process that brings information from the outside world into the body and into the brain, while perception is an active process that involves selecting, organizing, and interpreting information that's been brought to the brain by the various senses. Sensory organs begin the process by absorbing energy from a physical stimulus in the environment. Sensory receptors then convert this energy into neural impulses that are then sent into the brain. When the brain receives the neural impulses, it organizes the information and translates it into something meaningful. This is referred to as perception. The brain has the ability to discriminate between what is important and what is irrelevant, sorting interference from relevant input. We are constantly comparing information being received with what we have already encountered. How we perceive the world is a function of our past experiences, culture, and biological makeup. To view examples of perception, including the patterns of top-down and bottom-up processing, visit our Difficult Topics YouTube channel and view the video titled Top-Down and Bottom-Up Processing. Bypass Publishing presents Difficult Topics Explained. Psychology in the Fast Lane. Top-down and bottom-up processing. In talking about perception, top-down and bottom-up processing is a topic that's continually debated by psychologists and has been for a long time. How are sensation and perception related to cognition? Is perception constructed by cognition act to direct cognition? These are the questions that lead us to these different approaches. Let's start with bottom-up processing. Bottom-up processing is data-driven, and it states that perception directs cognition. That is, we start at the bottom with observable patterns, and these inform our higher-order cognitive processes. Now I can simplify this a little bit with an example. Sensation involves your eyes collecting visual information, your ears collecting auditory information, and so on with the other senses. So if you imagine a mosquito buzzing around your head, your eyes collect visual data about the surrounding environment, your ears hear the buzzing, and once this data reaches the brain and is processed, perception has occurred. The cognitive component then takes over, and the intention to swat the bug is formed and transmitted into action. So the bottom-up process says that this outside data is used by the brain's cognitive processes to do things like make decisions and determine behavior. Top-down processing. Have you ever woken up in the middle of the night and shuffled to the refrigerator or bathroom in the dark? Sure, all of us have. But this is where the concept of top-down processing enters the picture. This approach states that our behavior is influenced by conceptual data housed within the higher order cognitive processes that construct our experiences and expectations. So, put more simply, with an example, you're wandering around your apartment in the dark. You're able to get around fairly well because you've conceptualized what your apartment looks like in the light from previous experience. That is top-down processing, because your perception and behavior was driven by what you expected to see, not just by the visual data alone. So, bottom-up processing states that perception directs cognition or behavior, and top-down processing states that perception is constructed by cognition. Fast Lane Difficult Topics Explained. Psychology in the Fast Lane. Proactive and retroactive interference. Proactive and retroactive interference are both easy to remember if you draw a picture showing the relationship between old information and new information that you have stored in your memory. Synonyms for the word proactive are driven and forward-looking. So it's easy to remember that proactive interference is when old information moves forward and goes ahead to interfere with the retrieval of new information. An example. If you had learned tennis as a child, old information such as tennis rules and techniques might interfere with new information, like if you tried to learn racquetball later in life. Your old tennis information proactively interferes with the retrieval of your new racquetball information. Now the prefix retro denotes an action that is directed backward. So we can easily remember that retroactive interference is when new information goes backward into the past and interferes with the retrieval of old information. And to continue the example, now that you've just learned how to play racquetball, somebody suddenly asks you all about tennis rules, and it takes you a moment to think about it. This is retroactive interference because your new knowledge of racquetball is going backward to interfere with the retrieval of old knowledge of tennis. Bypass Publishing presents Psychology in the Fast Lane, Difficult Topics Explained. Schemas, Assimilation, and Accommodation. A schema is an organized pattern of thought that establishes a mental framework that represents some aspect of the world. We develop schemas for all types of items and activities, from simple items such as a chair, a car, fish, bird, or house, to complex like the chemical bonds between atoms or the seating in the House of Representatives. In short, we develop cognitive patterns for many things. We then use the schema we have developed as a means to compare new information against. Here's an example. You have a schema for a chair, a bed, and a sofa. The first time you see a futon, you immediately search your stored schema to see if it fits with anything you have already encountered. If it comes close, you make subtle changes to your schema to include the new item. In this example, you now have a schema of a sofa that includes the traditional characteristics along with those of a futon. So when you can add new information into an existing schema with little effort, you have experienced the cognitive process known as assimilation. When you need to store new information that conflicts with the schema you already have in place, a schema overhaul is in order. The process of accommodation involves the altering of schemas as a result of new information or new experiences, and new schemas may be developed during this process. Bypass Publishing presents Psychology in the Fast Lane, Difficult Topics Explained. Classical Conditioning, Everyday Applications. Classical conditioning associates a neutral stimulus with an unconditioned stimulus to produce a conditioned response. Classical conditioning can be used to explain many everyday applications. Sometimes people who are undergoing chemotherapy develop an aversion to foods that they've eaten right before their treatment. Chemotherapy naturally produces nausea, so chemotherapy is the unconditioned stimulus, and nausea is the unconditioned response. Right before arriving at the hospital, the person ate a strawberry ice cream cone, which has never made them ill in the past. So the ice cream cone is therefore a neutral stimulus. Through one trial learning, the person is classically conditioned to now feel sick whenever eating a strawberry ice cream cone. The ice cream cone is now the conditioned stimulus, and the nausea at eating the ice cream cone is a conditioned response. Bypass Publishing presents Psychology in the Fast Lane, Difficult Topics Explained. Reinforcers and punishers. A reinforcer is a stimulus or consequence presented after a response that increases the frequency of the response. For example, after a dog obeys the command to sit, he receives a reinforcer, like a treat or a hug or a toy. Eventually, the dog will be conditioned to sit by using reinforcement, where the treat and the hug and the toy are the reinforcers. Now, a punisher is a stimulus or consequence presented after a response that weakens or stops the response. For example, after the dog disobeys the command to sit, he receives a punisher. He'll get swatted, yanked, or yelled at. Eventually, the dog will be conditioned to sit by using punishment, where being swatted, yanked, or being told no are the punishers. Reinforcers and punishers usually fall into two different categories: primary and secondary. Primary reinforcers and punishers are stimuli that naturally trigger unlearned universal responses. Primary reinforcers are pleasant, satisfying things like food, water, and sex, and primary punishers are unpleasant things like pain and freezing temperatures. Secondary reinforcers and punishers are stimuli that we learn to respond to, and they're associated with or symbolic of primary reinforcers and punishers. Secondary reinforcers include things like money or good grades. Secondary punishers include things like a speeding ticket or withdrawal of approval. But the thing that reinforcers and punishers both have in common is that they're both presented to the situation to create some sort of change in the behavior or response. Bypass Publishing presents Psychology in the Fast Lane. Difficult topics explained. Negative reinforcement versus punishment. Many people confuse the terms negative reinforcement and punishment, thinking they mean the same thing, but they don't. Remember, reinforcers in general strengthen voluntary responses, and punishers weaken voluntary responses. So, growing up while I was in school, my mom could have punished me whenever I received bad grades in order to weaken whatever behavior was causing me to get bad grades. Or she could have reinforced my good behavior with something desirable whenever I did study or get good grades. Now, there are two types of each approach, positive and negative. Positive reinforcement is a process that strengthens the likelihood of a particular response by adding a stimulus after the behavior is performed. Negative reinforcement also strengthens the likelihood of a particular response, but by removing an undesirable consequence. So my mom could have told me that if I got good grades during the school year, then I wouldn't have to get a summer job, and that would be my reinforcer. Note that in both cases, the response of getting good grades is strengthened. We can punish a response by also adding or removing a stimulus. Like I could punish the bad behavior of a child by spanking its backside. Or I could punish you after you speed by taking away your driver's license. Both of these things would act to suppress certain bad behaviors. So punishment is either adding or removing something that would stop a response from happening again. And reinforcement is adding or removing something that would encourage the response to happen again. Bypass Publishing presents Difficult Topics Explained. Schedules of Reinforcement Applications. This segment is to simply give you some examples of the different schedules of reinforcement. We'll start with fixed interval. Fixed interval requires that you'll wait a specified period of time after completing the correct response before you'll be reinforced. An example of fixed interval is used by someone who gets paid every 2 weeks at their job. They have to show up to work and do all their assigned tasks, but no matter how fast they work or how many tasks they complete, they don't get paid until after the 2 weeks have transpired. Fixed interval schedules tend to produce overall response rates that are low and that increase as the time for reinforcement gets closer. This is called a scalloped response pattern. Now an example of variable interval. Variable interval would be, say, when someone waits for an elevator. Anytime they have to wait an unpredictable amount of time, they are on a variable interval schedule. People press the elevator button over and over again when they're in a hurry, but they only need to press the button once. It doesn't matter how many times you press it again, you just have to wait for it to arrive. Variable interval schedules produce steady rates of responding. Now for fixed ratio. Imagine you had a summer job where you stuffed envelopes and got paid for every 100 envelopes you stuffed. When you get paid for a set number of items you complete or sell, that's an example of a fixed ratio schedule. Fixed ratio schedules produce high rates of responding that decline immediately after the reinforcer is received. This is called a post-reinforcement pause. A classic example of a variable ratio schedule is known as the one-armed bandit, which is a slot machine. You must pull the arm an unspecified number of times before you are reinforced with a jackpot. Sometimes you can just put in a few quarters, pull the arm once, and win a big jackpot. Other times you pull and pull, but still never win. Variable ratio schedules produce high and steady rates of responding. Bypass Publishing presents Psychology in the Fast Lane. Difficult topics explained. Inductive and deductive reasoning. Oftentimes, we encounter seemingly contradictory information, such as nuclear power is good and nuclear power is dangerous. When faced with confident claims from both sides, it's often difficult to arrive at a conclusion as to which opinion is true. Deductive and inductive reasoning are two cognitive processes that we might use to arrive at a conclusion. Deduction is the process where a conclusion is drawn based on the acceptance of a number of premises or general principles. In other words, we first consider general information before we make specific conclusions. Consider a couple of really general statements such as monkeys like bananas and Lucy is a monkey, to arrive at the more specific conclusion, Lucy likes bananas. Deduction works well as long as the general information we use is true. When that general information is flawed, such that Lucy is an unknown species of monkey that does not like bananas, logical errors can occur. For this reason, our knowledge of nature cannot come from deduction. As part of the scientific method, researchers engage in inductive reasoning by conducting experiments to observe specific behavior before inferring general principles. In this way, we should observe Lucy and other members of her troop before arriving at the specific conclusion that Lucy likes bananas.
Bypass Publishing presents Psychology in the Fast Lane. Difficult topics explained. Theoretical approaches to psychological disorders. There are four generally accepted approaches to understanding psychological disorders: the biological approach, the psychological approach, the sociocultural approach, and the interactionist or biopsychosocial approach. The biological approach attributes psychological disorders to internal causes that are considered to be organic, such as brain injury, disease, or genetics. This approach would consider the cause of mental disorders to be a result of abnormalities in brain structure, imbalance in neurotransmitters or hormones, or genetic in nature. The psychological approach to understanding mental disorders uses all perspectives of psychology as the foundation for understanding the factors associated with psychological disorders. This approach would consider mental disorders to be a result of unconscious conflict, as in the psychodynamic perspective, the rewards and punishments an individual received, as in the behavioral and social cognitive perspective, or personal qualities and potential, and as in the humanistic perspective. The sociocultural approach attributes mental disorders to a larger social context, such as an effective functioning of the family, neighborhood dynamics, economic stress, ethnic impact, gender, and cultural values. Instead of focusing on the internal malfunctioning of an individual, this approach focuses on the larger environment involved in a person's life. This framework often overlaps with approaches seen in anthropology and sociology. The interactionist or biopsychosocial approach sees abnormal behavior as a combination of factors that may include the brain, genetics, cognition, self-esteem, family, and/or poverty. This approach may involve more than two frameworks already mentioned mixed with other frameworks coming from other perspectives and approaches. To locate more detail on any of these approaches, simply type the approach name into a web-based search engine such as Google, and you can find out just about as much information as you'd ever want.
Bypass Publishing presents Psychology in the Fast Lane. Difficult topics explained. Biological bases of emotion. Imagine watching a scary horror movie at home when the power suddenly goes out and there's a mysterious bump at the door. Such an event might elicit an emotional response like fear. One aspect of emotion includes physiological responses such as a racing heart rate and the hairs standing up on the back of your neck and things like that. Usually, these physiological responses are not voluntary because we don't consciously think, I want my heart rate to increase. The autonomic nervous system controls these involuntary physiological reactions to stimuli in the environment. Specifically, the sympathetic division of the autonomic nervous system releases hormones such as epinephrine and norepinephrine to cause a fight or flight response. During fight or flight, we may experience increased heart rate, blood pressure, sweating, and breathing, to name a few physiological symptoms, as we prepare to either battle for our lives or run away as quickly as we can. The sympathetic division of the autonomic nervous system allows the brain to direct the rest of the body. Within the brain, there are several brain areas involved in how we experience emotion. One important set of brain structures is the limbic system. The limbic system includes a number of structures such as the amygdala, hippocampus, hypothalamus, and regions of the cerebral cortex. The hippocampus is heavily involved in memory formation, and the hypothalamus serves to regulate motivated behaviors such as hunger and thirst. Research suggests that the amygdala is involved in assessing and interpreting situations to determine what emotions are appropriate, and it serves to connect these interpretations to the regions of the brain responsible for stimulating action. Bypass Publishing presents Psychology in the Fast Lane, Difficult Topics Explained. Schachter and Singer's two-factor theory. How do you feel? To answer this question, it's important to realize that emotions include cognition, feeling, behavior, and arousal. How these elements of emotion relate to one another has been debated by a number of theorists over the years. The cognitive arousal, or two-factor theory, proposed by Stanley Schachter and Jerome Singer in 1962, states that two things happen when someone encounters a situation that elicits an emotional response. First, physiological arousal occurs within our sympathetic nervous system. Second, cognitive interpretation of the arousal occurs so that we can identify the emotion we are experiencing. Imagine seeing a car crash on your way to work. Obviously, such an event could be traumatic. How do we, as human beings, identify the emotion that such an unexpected event triggers within us? If we rush to help the car crash victim, we might find that our heart and breathing rates increase. These are physiological responses produced by our sympathetic nervous system. The degree of physiological arousal will determine the intensity of emotion that we experience. During cognitive interpretation, we assess the situation and determine what emotion is appropriate by consciously linking the arousal we're experiencing with what is happening around us. Even though we were not directly involved in the car crash, we might find ourselves experiencing fear and/or sadness. Because we're all different people, situations or experiences are likely to affect us in very different ways. Appraisal refers to the cognitive act of assessing and evaluating the potential impact an event is likely to have on you. Primary appraisal is the initial step in appraisal where a person asks him or herself, "Is this a threat?" If the answer is yes, she will experience physiological stress, perhaps her stomach is knotting up, and an emotional reaction, perhaps anxiety or fear. During secondary appraisal, she has to determine how to cope with the threat. Initially, she may be worried about the life of the victim, and this may cause increased stress due to anxiety. She might catastrophize that the injured party will die, and having noticed a baby seat in the back of the car, a child will be orphaned. Reappraisal of this situation may occur if new information is encountered. For instance, hearing from a paramedic that the car crash victim will survive may assist coping and bring stress down to a manageable level. Bypass Publishing presents Difficult Topics Explained, Psychology in the Fast Lane, Top-Down and Bottom-Up Processing. In talking about perception, top-down and bottom-up processing is a topic that's continually debated by psychologists and has been for a long time. How are sensations cognition and perception related to cognition? Is perception constructed by cognition? Or does it act to direct cognition? These are the questions that lead us to these different approaches. Start with bottom-up processing. Bottom-up processing is data-driven, and it states that perception directs cognition. That is, we start at the bottom with observable patterns, and these inform our higher-order cognitive processes. Now I can simplify this a little bit with an example. Sensation involves your eyes collecting visual information, your ears collecting auditory information, and so on with the other senses. So if you imagine a mosquito buzzing around your head, your eyes collect visual data about the surrounding environment, your ears hear the buzzing, and once this data reaches the brain and is processed, perception has occurred. The cognitive component then takes over, and the intention to swat the bug is formed and transmitted into action. So the bottom-up process says that this outside data is used by the brain's cognitive processes to do things like make decisions and determine behavior. Top-down processing. Have you ever woken up in the middle of the night and shuffled to the refrigerator or bathroom in the dark? Sure, all of us have. But this is where the concept of top-down processing enters the picture. This approach states that our behavior is influenced by conceptual data housed within the higher-order cognitive processes that construct our experiences and expectations. So, put more simply, with an example, you're wandering around your apartment in the dark. You're able to get around fairly well because you've conceptualized what your apartment looks like in the light from previous experience. That is top-down processing, because your perception and behavior was driven by what you expected to see, not just by the visual data alone. So, bottom-up processing states that perception directs cognition or behavior, and top-down processing states that perception is constructed by cognition.